So in chapter 5, we're going to look specifically at the structure of the plasma membrane, and we're also going to look extensively at how things get across the plasma membrane, either coming into the cell or leaving a cell. And there's several categories of how things move across the membrane. So it depends mainly on size of the item that's trying to move across the membrane, and also whether it's a polar or nonpolar type molecule. And so we're going to start off with the structure of the membrane. And you'll remember that the basic structure of the cell membrane, and really all the membranes, even the ones inside of cells for eukaryotes, are fundamentally a phospholipid bilayer. So you'll remember that this is a phospholipid, one of them that I've circled, and they have this polar hydrophilic head, because that's where that glycerol in that phosphate group and then some other charged things are there. And then the fatty acids make up the tails, those are hydrophobic. So when you have lots and lots of these molecules, they will naturally assemble into what's called the phospholipid bilayer. The extracellular fluid is a water-based solution, so we call it aqueous. Because remember, water is always the solvent in biology. And the intracellular fluid is aqueous, water-based. And so what's going to happen, aqueous, and so what's going to happen is the heads of all of these many phospholipids will all face the water, and they interact quite happily with the water here. The heads of these phospholipids on this layer will interact with the water here, and then all of these tails are hiding in the middle here. They don't have to interact with the water, and that's what is very stable for them because they are hydrophobic. And so you end up with this hydrophobic layer or bilayer, but this section that's hydrophobic, and that's the barrier that keeps the aqueous solution that's outside the cell outside, and it keeps the aqueous solution that's inside the cell inside, because water cannot cross through that hydrophobic region. So this is the fundamental structure of the membrane, but it isn't the only thing that's in the membrane. So the full structure of the membrane is shown here. An artist has drawn this picture here. And you can see the phospholipid bilayer, and they're just showing a segment of the membrane, and they're also showing a slice through, so you can see inside the cell and you can see outside the cell at the same time. And so the phospholipid bilayer you can see here. Also in the phospholipid bilayer, there will be cholesterol. So I'm circling the cholesterol. They're showing it here as this little green thing. Cholesterol is very hydrophobic, so it's going to stay in that hydrophobic portion of the membrane. Now also, punching through the membrane will be these proteins shown in blue. If it goes all the way across the membrane and pokes out a little on both sides, those are called transmembrane proteins. And if they even partially go through the membrane, they're called integral. So integral proteins extend at least partially into that hydrophobic section. Transmembrane actually go all the way across the membrane, and they poke out on both sides. So those words work. And some proteins, though, are just sort of stuck on one side. They don't really extend into the membrane at all. Those are called peripheral. So you have these integral or transmembrane proteins that go through the hydrophobic section, and you have peripheral proteins that are sort of stuck either on the inside surface or on the outside surface. Also attached to the outside surface, you'll remember, the extracellular matrix proteins, which are collagen. So those are crossing on the outside surface. Those are fibers. And you also have these little proteins here that are called glycoproteins. So what they're showing here is you're seeing part of the protein sticking out of the membrane. So this is a integral protein that also has a little carbohydrate chain. You'll recognize the hexagons shown in green here. And so a glycoprotein is a protein that contains a little segment of carbohydrate on it. Similarly, a glycolipid is a molecule that it's really a phospholipid with a little attachment of carbohydrates. So that root GLYCO, glyco, refers to an attachment of a little bit of a carbohydrate, a little bit of a sugar chain polysaccharide there. So you have these glycoproteins with their carbohydrates facing the extracellular side of the membrane. Then you have proteins that go all the way across the membrane. You have cholesterol that's embedded in the hydrophobic section of the membrane. And on the inside of the membrane, that's where you have the connections to the cytoskeleton. So you'll remember the cytoskeleton contains these different filaments, microfilaments made of actin and intermediate filaments, for example. And so to some extent, there are connections between all of these things in various ways. And so this is called the fluid mosaic model of the membrane. It's just our current model, our current concept of how the membrane components are arranged. So just to go through this again, the main part of the membrane is the phospholipid bilayer. That's everywhere. It is very flexible. It has the consistency. It's, I like to think of like a soap bubble, like when you're blowing bubbles with a little wand and you blow a bubble. It's not quite that fragile, but it has this fluid ability to kind of move, without breaking. And so it's very flexible. The cholesterol is embedded in the hydrophobic section of the phospholipid bilayer. And then the proteins, if they go partially across the membrane, they're called integral. If they go all the way across, they're called transmembrane. And if they're only sticking on one side or the other, they're called peripheral. And so these proteins serve purposes. Some of them are just attaching to the extracellular matrix or to the cytoskeleton. Some of these proteins are acting as pores or channels for things to move through and to go across the membrane through these channel proteins. And the ones that are the glycoproteins and the glycolipids, these are for the immune system to be able to recognize which cells belong to you. So in other words, your immune system will scan the outside surface of all your cells and make sure that the glycoproteins and the glycolipids are the ones that are expected to be there. So early when you're very young, your immune system learns which glycoproteins and glycolipids are on your cells. And after that point, it will scan and make sure that's the only ones that are there. If it finds a cell that has a glycoprotein or a glycolipid that does not belong to you, it will kill that cell. It will attack. And so that's fundamentally why if you get a transplant, like an organ transplant, they say you have to match, you have to find a donor who has a match. And what that match means is they have to have the same glycoproteins and glycolipids that you do. If they do, then when your body is scanning that implanted organ, it will think that those are the same glycoproteins that you have and it won't attack it. But if the organ transplant is not a match, the immune cells will eventually kill the organ that has been transplanted. And we call that rejection. So those cell surface markers are important, but we're not going to talk about them too much more in this chapter because what we're really talking about in this chapter are these transmembrane proteins that allow molecules to go from the outside of the cell environment into the cell or from the inside of the cell out. So we'll be talking about those transmembrane proteins quite a bit. The way that scientists actually figured out how the plasma membrane was structured is an old technique called freeze fracture. They could actually freeze cells think about like liquid nitrogen. They would freeze cells, very deep, deep freeze, and then they could use a blade to separate the two layers of the phospholipid bilayer and look at them under the microscope. And from doing this, they figured out quite a bit about how the structure of the membrane was set up. So they learned a lot about the fluid mosaic model from that information. And they also did an experiment. This is a fun experiment to think about, and this was done very early on as they were studying and learning about the membrane structure. What they did was they took a human cell and they used a fluorescent stain to mark the proteins that are in the membrane of that human cell. Then they took a mouse cell and they used a red stain to label or to stain the proteins in the mouse cell. So these are those transmembrane proteins. And their question was, are the components of the membrane able to like move around, or are they fixed in place? Because when you do a freeze fracture, everything appears to be fixed, but that's because it's frozen. So in a real cell, are the components able to move? And so what they did was you can disrupt two membranes and force the two cells to fuse together. And so that's what they're showing you here. They fuse those cells together, a human cell and a mouse cell. Now, guys, these aren't eggs and sperm. They're not making some kind of weird embryo here. These are just two cells. And when they fuse them together, at first, half of the new fused cell would have the blue proteins on that hemisphere, and the other hemisphere of the new cell would have red. But they came back later and they found that the cells that had been fused together now had all of these proteins mixed. That means that that's indirect evidence that the different proteins can move around in the membrane, right? So we call it a fluid membrane. So part of the fluidity of the membrane has to do with what kind of fatty acids are in the phospholipids. If cells use very saturated fatty acids to make their phospholipids, then their membranes will be a little less fluid, not stiff, but a little bit less fluid than if they use the unsaturated fatty acids. So cells will adjust which fatty acids are being used to make phospholipids depending on what kind of environment they're in. If it's very warm outside, the membrane gets too fluid, then the cell will adjust by making the membranes a little more stiff, a little less fluid, a little more rigid. If in cold temperatures the membranes are getting too stiff, it can use some unsaturated fatty acids to make them a little more fluid. Don't get too tied up in that, but the idea is that the cell can adjust and keep the fluidity of the membrane just right by adjusting how it makes the phospholipids that it is making and putting into its membranes. But this experiment showed that the components of the membranes, including the proteins in this case, can move around in the structure. And so that's part of why we call it the fluid mosaic model. So membrane proteins, just like any proteins, membrane proteins have a lot of functions. Cells depend on their proteins for everything. And so in this case, we're specifically focusing on the proteins that are in the membrane. Those proteins, we're going to focus quite a bit on them being transporters in this chapter. So put a star by that. Some of the proteins that are in a cell can be attached to membranes, so they can be enzymes and membrane proteins at the same time. I already mentioned to you the proteins that are cell identity proteins are called glycoproteins. In chapter 4, we looked at junctions. Those are proteins that hold cells together and in some cases act as passages between two cells. They can connect to the cytoskeleton. They can act as receptors to receive chemical messages from the external environment, from the extracellular environment. But in this chapter, we're really going to focus on this transport function of membrane proteins. So on the next slide, these are pictures relating to those functions on the last slide. But this one is the one we're looking at, the transporter concept. Some proteins that are embedded in the membrane, and you can see they've turned this membrane sort of in a vertical way here. and then you're just looking at a section of the membrane, and then they've sliced through it, and so you've cut through one of these channel proteins shown in blue. We're going to learn about the rules that govern how different things can move into a cell in this drawing or out of a cell, either way. So that's the main function of the proteins that we're going to be studying in this chapter. All right, the proteins that cross the membrane, they're called transmembrane proteins. They have regions or domains. Now, when I say domain in this context, I do not mean archaea, bacteria, eukarya. That usage of the word domain is about classification. In this context, when I say domain, I'm just talking about some section of a protein. And there are parts of proteins that are very well structured to go across that hydrophobic section of a phospholipid bilayer. And in this drawing, they've shaded those in blue. And very commonly, those are what we call alpha helix, which is a secondary structure of protein. And they can also be beta sheets. So you often see a spiraling section of protein as it crosses over a membrane, or a beta sheet we'll see on the next slide. So different transmembrane proteins have different details of their structure, but it's very common to see these spiraling or beta sheet structures that cross the membrane. And then you have some of the proteins sticking above the membrane here, and some of the proteins sticking down below. So this is truly a transmembrane protein. This is a transmembrane protein, but it actually goes across the membrane many times. So you cross the membrane, it looks like 7 times. This is probably a G protein. 1, 2, 3, 4, 5, 6, 7. So 1 time across the membrane, 7 times across the membrane, they're all called transmembrane proteins. And some of these create pores, which molecules can pass through. So a good picture is here. This is a pore protein. Again, they're showing you a slice through a membrane, and then they're showing you the whole pore protein. So if you look at it, it's like a barrel. They're trying to give it to you almost in a 3-dimensional way, but you're looking at the barrel from the side. So if we could go above the picture and look down, there would actually be a hole in the middle of the barrel. All right, sometimes this is called the beta barrel. And so a lot of pore proteins have this shape, the beta barrel. And the objects or the molecules, the particles can go through that hole. But you can imagine that the hole has a certain size, and so it can't allow just any size molecule. These are going to have to be fairly small molecules that are going to be able to go through these pore proteins. Okay, so that is a general introduction to the transport proteins that we're going to be talking about, these pore proteins. In the next section, we're going to talk about some of the rules that govern which way molecules will move.